Thank you very much to ILSI for putting together this wonderful session this morning. It's a pleasure to be here. Thanks. Well, we've gotten an idea now of the different types of measurement error that we see in nutritional epidemiology studies. On one hand, we may have false positives due to multiple testing. On the other hand, we may underestimate our results based on attenuation due to imprecision and random error. What I'm going to do is talk a little bit more about food frequencies, but first there's my disclosure slide that is required, and go through what we really need is good usual intake at the individual level. There's been lots of work in different ways that people have tried to look at different instruments to do statistical correction. We've heard about random error, we've heard about person-specific bias. I'm going to introduce a new bias, which is subgroup-specific bias that I've been dealing with that I think is really creating quite a big problem for current work in nutritional epidemiology. So we need valid and reliable nutrient intake, but the truth is we're not just dealing with a uniform population in the United States or in the world. At the NIH, most of you know you need to include minorities, you need to include men and women, different age groups. We are getting more and more complex studies where we're having pooling projects and trying to put data together. We're looking at gene-diet interactions where we're really trying to put individuals in cells. And so no kind of overall statistical correction is going to be able to place the individuals in the correct cells in order to do those interaction analysis. We just haven't gotten to that point yet. So I'm going to argue that all this statistical correction is great and it can help when you know your population and you know your data, but that in fact what we really need is much more work to get true individual usual intake in order to solve this problem. But as Kevin pointed out, it's not done right now because it's very expensive. And it takes a lot of effort that most studies have not been willing to accept, either the cost, the time, the participant burden. And so it's a huge challenge that we have to face right now. So dietary intake is our primary exposure in nutritional epidemiology. We have the different instruments that you've already heard about, but the truth is that as soon as you make an instrument like a food frequency questionnaire, it's out of date. The current food frequency questionnaires don't have energy drinks on them, for example, or different kinds of bars. The diet is constantly changing. It's complicated. And those food group levels will give you, as Kevin alluded to, or others, qualitative information that can be very useful. We look at dietary patterns, we look at food groups, and they can tell us a lot. We can tell you that eating fruits and vegetables is good. That's been very well replicated. But as we're getting into more complicated issues, especially with gene-diet interactions, we're sometimes looking at specific biochemical pathways. We need to know exactly about vitamin B6 in that pathway. We're not going to get that with the current instruments. So we really, really need to work on that. Kevin's gone through the methods, so I think I will pretty much just skip through this slide, except to note that we are often needing to focus on long-term usual intake. And just to go back to the point that was raised about lack of reproducibility, I do want to caution that sometimes we do have good evidence in epidemiology studies, and then when they do a clinical trial, it doesn't pan out. But you have to be careful not to say that that means the epi studies were wrong, because long-term usual intake, as Kevin mentioned, exposures over your lifetime may condition a risk for heart disease and cancer, and a 6-month or even a 2-year trial with a specific nutrient may be too little, too late, or too much too late in the case of the beta-carotene studies. So it's much more complicated than saying the clinical trials are always right. It's a matter of putting everything in context. But What I've been dealing with as I've been working with minority populations and health disparities is realizing that the instruments we have available for use in those populations are very inadequate, probably wrong in many cases, and that it's not just random error. It can actually be biased, particularly when we look at studies like MESA, where there's multi-ethnic groups. They're all using the same instrument, but the instrument really was designed more for the general population with a few foods added in for the other groups. So it's not measuring everybody's intake with the same precision. It's got differences in the biases depending on the groups below that. So we have to worry about that in terms of looking beyond just measuring and adjusting for day-to-day variation. Day-to-day variation will adjust for your whole sample, but within the sample, for example, day-to-day variation in rice intake is going to be much different for a general population than it is for an Asian population or for the Puerto Rican population, where that ratio will be quite different. And we're just looking at the average of the whole thing when we do this. And so it does differ in the precision for those subgroups. And we are interested in the subgroups more and more as we look at health disparities and differential risk of disease, and particularly when we put that together with genetic risk. So, again, the de-attenuation methods are very useful, but they're not, as Kevin said, widely used, and even if they are used, they're not solving the problem. So Kevin introduced the NCI approach, where they add a propensity questionnaire to 2 24-hour recalls to adjust for infrequently consumed foods. And this is one of the best methods we have out there. Again, it's not that widely used outside of the NIH yet, but it is a good method. But still, it's not dealing with the differences across the subpopulations, and it's still not getting, the important thing to realize is that it's still not getting to the individual intake, because they can adjust, say, for liver, and they can know that it may not appear in 2 24-hour recalls because someone may eat it only once every 2 months, and you've got that information, then you can adjust for. But that's still not putting that vitamin A into the cell for the person's vitamin A. It's adjusting for it after the fact. So it's getting to usual intake distributions that it can improve estimates of association, but it's not able to deal with individual specific intakes that can then be put in a cell that they have a low intake or a high intake that may interact with a genetic polymorphism, for example. So we haven't gotten there yet, and it's a real problem as we're doing more and more of these kinds of studies. So what do we know about what we're doing right now in multi-ethnic populations? First of all, the day-to-day variation can no longer be assumed to be equally distributed throughout the population. The propensity questionnaire is not adequately specific for diverse groups that may eat something almost every day that's not even on the questionnaire if they come from Cambodia, for example, and have a different diet. And we certainly find that, for example, with the Puerto Ricans that I work with, is plantains, for example. It's a very commonly consumed food, not on any of these questionnaires. So you've already got a big piece missing. This is really something that needs a lot more attention. The limitations of the existing statistical corrections, again, just to reiterate, they don't consider the major dietary pattern subsets, including, I was just out at the International Vegetarian Conference in Loma Linda. There's a lot of things that vegetarians eat that are not on these questionnaires. And if I administer questionnaires, I often get people asking me, where do I put some of my soy products, for example, that aren't on the questionnaires? Because the questionnaires are designed for the majority population. So what this can do is it can not only lead to attenuation, but it can lead to systematic bias by subgroup. It can also lead to systematic bias and changes over time, depending on how the questionnaire is designed and what it's missing. Food frequency questionnaires, as noted, are the major tool that we use in nutritional epidemiology, used by the Harvard School of Public Health, the Framingham study, most of the big studies, because it simply is the most efficient way to get dietary intake. It's considered semi-quantitative. In use with the populations for whom it was designed, and I will say, for example, the nurses, they have a limited range of dietary intakes for the majority. And so they have been able to rank individuals on different dietary intakes, nutrient intakes, and dietary patterns, and find a lot of important things. However, when that questionnaire is expanded to other groups, including, let's say, African Americans in the South, and I will show you some data from the Jackson Heart study that I work with, it doesn't work so well. And so we need to look at that more carefully. I will also just mention the open study, which we heard about, that have highlighted the attenuation of the food frequency questionnaires and their lack of necessarily validity for energy and protein. However, at the same time, when we compare micronutrients, sometimes the food frequency questionnaires show better validity than a few recalls will. So none of this is simple. There is no right answer. The bottom line is we need a lot more work. So how are food frequencies designed? Well, basically, we take national data or data from the group of interest, and we look at the major foods contributing to nutrients. And if we do that for the whole population, we get a good estimate for the whole population. And the concept is, well, if there's one person here or a few people there that eat outside of that pattern, it won't matter. It leads to some error. It leads to some loss of power. But it'll all be able to get worked out in the end. However, as I've noted many times now, that's not necessarily true if you have a large subset that wasn't specifically identified and ranked, and so their foods fall out. The other thing that limits the power of the food frequency questionnaire are the compromises that are made, because we can't have a questionnaire with 2,000 foods and have people going for a whole day answering every single food. So we group things in line items. So other fruits other than apples, pears, bananas, citrus, melon, or berries. Behind that, we have weighted nutrient values for the foods based on their proportion consumption in the population. So again, this is going back to the average. We've got the weighted average of grapes, plums, peaches, pineapple, mango, and kiwi possibly in our other fruit category, and they would be weighted based on the majority population. Well, what does this do? Again, it is taking away from the very variation that we may really need. If you are someone in the group that eats a lot of mango, your vitamin A is going to be really underestimated. And if you're someone that eats a lot of pineapple, for example, you'll have a different nutrient. But everybody gets the same nutrient values for this, because that's the best we can do right now. So we're losing variation. Sometimes it's more important than others, but it can be very important, particularly when there are diverse cultural diets that sometimes eat some of these foods every single day, and they're being averaged together with all these other things. Another big problem that we have with the food frequencies when we use them with multiple groups is the assumption of standard recipes. And so this was another thing we really had to go through with the Puerto Rican data, because we realized that the existing food frequencies, not only were missing foods, but we're going to totally underestimate rice, which is eaten in much larger portions daily, and other things. So a weighted average of canned chicken soup for the full Hanes sample would be about half a cup to 3/4 of a cup, usually a can of chicken noodle soup that is assumed in the food frequency, but in Puerto Ricans, they would eat a very large bowl of chicken soup as a meal, probably with a whole chicken leg in it, and so it would be a completely different item, yet they would answer it the same way on the questionnaire. So this is going to lead to important bias. Just some examples of what's out there with people, or with studies that have tried to look at multi-ethnic groups. Just to give you an idea of what is happening. These are validity coefficients for the IRIS study. And here we're looking at non-Hispanic whites, urban African Americans, and rural Hispanics. And what you can see in yellow is that the validity coefficients are much, much lower, sometimes half or less, for the African Americans and for the Hispanics when compared to the non-Hispanic whites. So what does this mean? This means there's clearly a differential precision in the measurement for these different subgroups, and what that means down the road for our analyses will depend on what the specific analyses are, but it's clearly problematic. Just another one, this is the multi-ethnic cohort in Hawaii and Los Angeles. Again, you see the non-Hispanic whites at the bottom have respectable validity coefficients, but when you look at African Americans, and most people don't think that it should be so much different for African Americans, but you see it goes for energy from 48 to 16, for fat from 57 to 29. So there are very dramatic differences in this, and again, here with Japanese Americans as well. And just the last one, the Block and Harvard Food Frequency Questionnaires in the WIC Dietary Assessment Validation Study, where they did a validation study. Again, you can see that for non-Hispanic whites, the validity coefficients were much, much better, and they were really quite terrible for the Hispanics. And in the report on this, Gladys Block actually said that the Hispanics, it just didn't work for the Hispanics, this particular questionnaire. So that's a lot of work to have data that's so disappointing. And so what I realized when I started working with the Puerto Rican study was that the questionnaires that we did have were not going to work. They were missing foods, missing recipes, missing portion sizes. So we went out to create our own questionnaire. Our first study, we did use 24-hour recalls because I truly believe that in any new group, you have to start there in order to really know what they're eating. But then because we were doing a much bigger longitudinal cohort study and couldn't afford the multiple recalls, we did design a food frequency questionnaire to be used with them. And here's just an example of some of the things. This is a minor example, but there are others that have more of an effect. The food weights behind the items, and we started with a block questionnaire, and you can see that the assumptions behind that are that the American public's eating mostly kidney beans, pinto beans, and baked beans. But in the Puerto Ricans, they're eating more pink beans, and they're not eating baked beans. So all the sugar and the fat that would be in those baked beans is not part of their diet. On the other hand, when they cook their beans, they cook them with tomato sauce, and that's totally missing from this. And we validated that by looking at plasma lycopene and found that really, if you don't consider that tomato sauce, you've totally misestimated their carotenoids. These are just some of the foods that we needed to add that would have been missing. The plantains, as I mentioned, were a really big issue in terms of underestimation. This is how we looked at the portion sizes. We took all the foods that were major contributors on the 24-hour recalls and compared them between the Haines 2 and the Hispanic Haines. And I don't know if you can see this here, but this is the modal portion size for the non-Hispanic whites in the Haines 2, and this is the modal portion size for the Puerto Ricans in the Hispanic Haines. So we're talking about a difference of 200 grams to 500 grams. And if it's something that's eaten occasionally, that's not a big deal, but if it's something that's eaten weekly or more, it really does matter. We, of course, validated our tool after we looked at it and showed that if we use the original block questionnaire, we were certainly underestimating energy for this group. Once we revised it, we got more foods in, we did better. And once we added the right portion sizes, we got back close to the original values. This was true, of course, once it's true for energy, it's true for all the different nutrients, but you can see the dramatic underestimation of protein here by using these questionnaires. And so, in retrospect, this seems obvious, but the truth is there are a lot of studies that are using the block and the Willet Questionnaire with multi-ethnic samples and don't have this kinds of information. Even more important, when we looked at using the original block with the Puerto Ricans and a comparison of non-Hispanic whites, what we saw was a significant difference in the folate intake. So what you might report is that Puerto Ricans have significantly lower folate intake than non-Hispanic whites, although in the recalls we didn't see that at all. So once we did all our corrections, we were able to get it back up close to that. So the important point is here is that you can have a very valid, good food frequency that can work with the population that you're working with. It takes a lot of work, and it takes updating, it takes monitoring, and all the populations that you're interested in within the group that you're studying need to be included. So it's not simple. It's not simple at all. Just to move to another one, the southern U.S., we realized after talking with individuals in the Mississippi Delta, where the USDA had a big project, the Delta Nutrition Intervention Research Initiative, that their foods are not those that are typically on our standard food frequency questionnaires either, and they're a big group. And so we started working in the Delta region, led by Margaret Bogle. They did a big 24-hour recall survey of that region to find out what people were indeed eating. And based on those recalls, we found that there were lots of foods that needed to be added. So this is our whole list. We created, with Margaret and others there, we created quite a long food frequency questionnaire that would include the general foods existing on them, but also the southern foods. And they are not just a small number of things that need to be added. Things like grits and biscuits are obvious, but they're not on the Willet Questionnaire. So we needed to look at that. The other thing, even in the southern group, the portion sizes were much larger for several key foods than were on the block questionnaire. In the Willet Questionnaire, there are no portion sizes. You're expected to kind of multiply times a half cup or whatever as a reference. And that's a lot of what the underestimation is that results from the Willet Questionnaire and why it gets better when you adjust for energy. But in the block, they have small, medium, and large, but even the large was too small. So we expanded it to 4 portion sizes so that we were able to capture the full extent of that. That questionnaire has been working pretty well in regions of the South, and a lot of different groups are now using it. But based on that, I was asked to work on a questionnaire for the Jackson Heart Study, and this is where I also want to show you how important it is to invest in what you really want to get out of your dietary intake. So the problem with the Jackson Heart Study, we went down, we did some more qualitative interviewing, some more recalls in the area, talked to individuals, and found that the Delta NIRE questionnaire pretty well covered Jackson, Mississippi, and so we were ready to go with that. But the scientific board of the Jackson Heart Study said, no, that takes about 45 minutes to complete. We've only got 20 minutes for our patients to answer this questionnaire. We can't increase the respondent burden. And we get this all the time. We get this at Framingham. Tufts started putting the Framingham food frequency questionnaire in. There's always a competition for time. There's a competition for dollars in these big studies. They're often led by MDs who have other priorities than diet. So we said, how are we going to do this? And they were firm. We didn't want to exclude any foods, so we collapsed items so that we could be inclusive and make sure at least that everything was able to be included, and we wouldn't be losing total energy. But this is some of what we found. We've done pretty extensive validation of this. We actually found that by collapsing the items, we got pretty good results for energy, protein, macronutrients, because they were all in those categories. But as I showed you before, within a category, when you collapse for convenience, you've got weighted specific foods that have to be based on some kind of assumptions. And as you can imagine, what happens is that you lose it on the micronutrients. So here we've got differences, validity coefficients for the long full food frequency were pretty good. 73 here for iron, but these all went quite down for the short food frequency questionnaire that we are using. Not terrible, except we are losing a lot of information. I'm giving you a few examples here, but carotenoids is another one where you really lose tremendously if you don't have the specific fruits and vegetables. So, what are we going to do? There's more and more studies that are pooling data, combining data. Gene-diet interaction studies are struggling with how are we going to get good answers if we don't have good dietary data. There's no way that I can see around fully investing the time to realizing that there's no shortcut that I know of. Food choice is a very complex thing. It does vary tremendously in different subgroups, be they vegetarians, Latinos, Southern African Americans, others, Asian groups. And if the foods aren't there, you've got it wrong. Recipes are another huge issue in the Puerto Ricans. They eat rice every day and they cook it with oil all the time. We got all that information from our original recalls. If you use the assumptions in a regular food frequency questionnaire, a serving of rice is a half cup or 2/3 of a cup, and there's no oil. So you're underestimating both energy, fat, and refined carbs. So we need to get this detail somehow. Just to show you a little bit, one slide on interaction of SNPs of genetic polymorphisms. What we see here is some data from our Puerto Rican health study where we were able to show that high fat intake changes the relationship of the polymorphism to, in this case, waist circumference and blood pressure. And these are very important things as we move towards understanding health disparities and how different groups may be at different risk. But we wouldn't be able to get this if we have the fat misclassified, if we have people in the wrong level. And so we have to have that individual information. This is important just to emphasize that there are different genetic profiles of groups, and this is a study by Josie Mattei, who was one of our graduate students, who showed that protective minor alleles for lipid metabolism genes are less frequent and risk alleles more frequent in the Puerto Rican population than a non-Hispanic white population. So these are important questions, and if we're going to move the science forward, we really do need to get there. So what do we got? We've got the FENCS is a group that is trying to look at this. They're trying to get a toolkit of measures that can be common across studies so the studies can be pooled or work together in order to see the effects of gene-diet interactions or gene-environment interactions in lots of different groups. They struggled with this, and their current recommendations are to use the 24-hour recall, but then to use screeners such as fruits and vegetables, dairy, added sugars, calcium, et cetera. This may be the best we have right now because there's certainly not a food frequency that'll cover everyone, but it's not going to get us where we need to go. So, what can we do? Well, I think we have to look at technology, and the NCI and others have been funding some studies to improve dietary assessment, and I think, as was Kevin mentioned, there's efforts with photography, with web-based methods. None of them are there yet, but we need all the bright minds that we can get to try and think about how to do that. And as technology is getting faster, we can include longer questionnaires with skip patterns, perhaps, and use photography to capture things if we can make it more feasible. Some people have it hanging around the neck and taking multiple. I know Lenore Arab is looking at something that takes constant photographs. Lots of good ideas, but still a lot of work to do. So in summary, there doesn't seem to be an easy answer. We've really got to measure dietary intake at the individual level. There's no statistical magic that's going to take care of that. And we need to convince funders, reviewers, scientists to value the importance of comprehensive dietary assessment. We have an epidemic of obesity, diabetes, and diet-related diseases, and we can't ignore this issue. So I think it's up to all of us to communicate it and to work to get better technology to try and answer the question. Thank you.